And welcome to your asynchronous lesson on starting to develop your experimental design and question for our rock candy lab. Now today we did part one where we really identified the different candy stages in terms of temperature, percent sugar, and texture. And we also investigated with our observations in these two articles how the crystal structure or the structure of igneous rocks and candy really vary based on ingredients, temperature, environmental settings, and how those three factors sort of come together. Which is going to bring us to part three, which is all about being able to plan an investigation. Now, you and a partner or a small group are going to be investigating how chemical composition and factors influence crystal structure in candies. You can influence the composition of your lava by using various amounts of table, brown, sugar, corn syrup, and honey. You can influence your structure by taking samples at different temperatures and allowing them to cool at different temperatures. And that is part of the environmental variation as well. Now you're going to be plating these samples on microscope slides like we practiced in class today. And we want to make sure that they're as small and flat as possible so we can see them on a microscope for our analysis. Now the first thing that I have to note is that you're going to be using 4 different types of sugar that have different percents of water by mass. And then when it comes to their sugar component, they have various amounts of sucrose, fructose, and glucose. Sucrose being the thing that crystallizes to form different types of candy, fructose and glucose being additive or interfering agents that prevent sugar crystallization. So the first thing that I want each of you to do today is to develop a question. Thinking a specific scientific question related to crystal structure of candy related to the variables discussed above. So a great question could be, how does different amounts of fructose influence crystallization of sucrose at 250 degrees? Another great question would be, how different are sugar crystals from samples of table and brown sugar at all 6 stages? You're really just thinking about what is your dependent, which is crystal formation, and your independent, sucrose, fructose, temperature, stages, samples in your experiment. After you've developed a question, I want you to really think about your hypothesis, if then because, and this should refer to information that you can find in your information here. So I might say, if the development of sugar crystals is slowed with more glucose, then I would expect to see smaller sugar crystals in a solution with higher amounts of sucrose due to the interfering nature of it. So again, glucose and fructose sort of act like inhibitors. They break up the rave. They prevent the sugar crystallization from occurring when our solution's set. You're going to want to specify what your dependent and independent variables are. Your control group is going to be the sort of normal environment or the treatment / replicate / data point that we compare to. So you really want to think about what's your question and how are you going to compare that to what we think is normal. It might be sugar sample at 250, or it could be all sugar samples, or it could be white sugar samples only. You really want to think about what is the thing that I'm going to be comparing my sugar crystal formations to. Now, when it comes to developing your set of treatments, this is really want to start thinking about what are your environments. So it might be helpful to start thinking about how much sugar is going to be in each solution. So maybe I have 100 grams of solution. Maybe one is 100 grams of sucrose or table sugar. The next one is going to be 80 grams of sucrose and 20 grams of honey. So really starting to think about what are the different beakers of sugar and water that I'm going to be boiling to collect samples from. One tidbit is the smaller your solution is, the easier it will be to boil and collect samples, because the bigger your sample is, the more water we have to boil out, and that takes time. Your treatments should be based on your hypothesis. So if your hypothesis in question is based on sugar crystals related to sugar type, you should be modifying different treatments based on the types of sugars in there. And then in small bullet points, I really want you to think about your experimental procedure in terms of steps, constants, and data measurements. I sort of highlighted today how you would collect data and how you would sample data. So really think about the things that you want to write down in order to look at and refer to and think of conducting your experiment in a seamless manner. So maybe constant might be take 1 milliliter of syrup.
All students taking the bus, you are now dismissed. All students taking the bus, you are now dismissed.
You had to take the bus. I hope you are leaving now. Take 1 milliliter of syrup per sample.
All students taking the bus, you are now dismissed. All students taking the bus, you are now dismissed.
Interesting. I repeated. Place sample in center. Maybe let cool for 15 minutes. You really want to think about what are the things you're going to do the same every single time to make sure you don't introduce any unknown error. We will be workshopping these and talking about these on Thursday in class, but I really would like for you to finish this before, I see you on Thursday. I hope you have a great day, and see you soon.